Today we are going to learn about types of receptors. Please subscribe Medi queries for to learn more interesting concepts. Receptor protein. Receptors are the sensitizing elements in the system of chemical communication that coordinates the function of all the different cells in the body. The chemical messengers being the various hormones, transmitters and other mediators. Many drugs act either as agonists or antagonists on the receptors for non-endogenous mediators. Types of receptors. So based on the molecular structure and the nature of the linkage transduction, we can distinguish 4 receptor types. First one is ligand-gated ion channels, G-protein coupled receptors, kinase-linked receptors, nuclear receptors. Ligand-gated ion channels. So these are also known as ionotropic receptors. These are membrane proteins with similar structure to other ion channels but incorporate a ligand binding site in extracellular domain. These are the receptors on which fast neurotransmitters act. Example: GABA receptors, glutamate receptors, kinase receptors, glycine receptors. Molecular structure. The ligand-gated ion channels have structural features in common with other ion channels. The nicotinic acetylcholine receptor. It is a assemble of 4 different types of subunits termed alpha, beta, gamma, delta. The 4 subunits forms a membrane spanning alpha helices. The oligomeric structure possesses 2 acetylcholine binding sites. So each subunit spans membrane 4 times. So the channel comprises not less than 20 membrane spanning helices surrounds a center pore. The 2 acetylcholine binding sites lies on the N-terminal region of 2 alpha subunits. When acetylcholine binds, the kinked alpha helices swings out of the way, thus opens the channel. So here it is a ligand-gated ion channel while it is in open and in closed positions. So here is a top view. The gating mechanism. The receptors of this type controls the fastest synaptic events in the nervous system, in which a neurotransmitter acts on the postsympathetic membrane of the nerve or muscle cells and increases its permeability to the particular ions. Acetylcholine at the neurotransmitter junction for glutamate in CNS causes an increase in sodium and potassium permeability. This results in a net inward current carried mainly by the sodium ions, which depolarizes the cell and increases the probability that will generate an action potential. The action of the transmitter reaches a peak in a fraction of milliseconds. And the next category is G-protein coupled receptors. So the G-protein coupled receptors are also known as metabotropic receptors or 7 transmembrane spanning heptahelical receptors. So they are the membrane receptors that are coupled to the intracellular effector system via Z-protein. Example: muscarinic acetylcholine receptors, adrenoceptors, dopamine receptors, 5-hydroxytryptamine receptors, opiate receptors, receptors for many peptides, purine receptors. Molecular structure. Its structure comprises 7 transmembrane alpha helices with an extracellular N-terminal domain of varying length and an intracellular C-terminal domain. GPCR are divided in 3 families. They have the same heptahelical structure but differ in length of N-terminal and location of agonist binding domain. Family A, rhodopsin family, it is the largest group comprising a monoamine and a neuropeptide receptor. Structural features: it has a short extracellular tail. Ligand binds to the transmembrane helices or to the extracellular loop. Family B, secretin or glucagon receptor family, receptors for peptide hormones including secretin and glucagon calcitonin. Structural features: it has an intermediate extracellular tail and incorporating ligand binding domain. Family C, metabotropic glutamate receptors, GABA B receptors, calcium sensing receptors. Structural features: long extracellular tail incorporating ligand binding domain. Z protein and their role. They go between proteins but were actually called Z protein because of their interaction with guanine nucleotide GTP and GDP. G protein consists of three subunits, alpha, beta, and gamma. Guanine nucleotide binds to the alpha subunit, which has enzymatic activity, catalyzing conversion of GTP to GDP. The beta gamma subunits remains together as beta gamma complex. In resting state, the G protein exists as alpha beta gamma trimer with GDP occupying the sites on the alpha subunits. So when a G protein coupled receptor is occupied by an agonist molecule, it acquires high affinity for the alpha beta gamma trimer. Association of the trimer with the receptor causes a bound GDP to dissociate and to be replaced with GTP. Which in turn causes dissociation of G protein trimer, releasing alpha GTP and beta gamma subunits. These are the active forms of G protein which diffuses in the membrane and associates with various enzymes and ions channels, causing the activation or inactivation. The process is terminated when the hydrolysis of GTP to GDP occurs through the GTPase activity of the alpha subunit. The resultant alpha GDP then dissociates from the effector and reunites with beta gamma complex, completing the cycle. So the attachment of the alpha subunit to an effector molecule increases its GTPase activity. Next category is type 3, kinase linked receptors. Receptors for various hormones, example insulin, growth factor, incorporate tyrosine kinase in their intracellular domain. The receptor possesses a large extracellular ligand binding domain connected via single alpha helix to the intracellular domain. They are mainly involved in events controlled cell growth and differentiation. They also act indirectly by regulating the gene transcription. Two important pathways are the Ras-Raf-MAP kinase pathway, which is important in cell division, growth and differentiation. JAK-STAT pathway, which is activated by many cytokines and which controls the synthesis and release of many inflammatory mediators. Next category is nuclear receptors. Ligand includes steroid hormones, thyroid hormones, vitamin D and retinoic acid, lipid lowering and antidiabetic drugs. Receptors are intracellular proteins, so ligand must first enter cell. Receptor consists of conserved DNA binding domain attaches to the variable ligand binding and transcriptional control domains. So DNA binding domains recognize specific base sequence, thus promoting or repressing particular genes. So effects are produced as a result of altered protein synthesis and therefore are slow in onset. This is about receptors. Thank you for watching this video. Please subscribe Medi.